So the agenda for today, first, our two main assays for the Tapestri platform is a DNA-only workflow in which you use targeted NGS panels where you profile for mutations, or you can say you're genotyping, whereas our additional workflow where you can, on top of profiling for genotype information, you can use a set of surface markers and where in which you conjugate a tag sequence to antibodies that will bind to your surface expressing proteins with a panel of 15 to 20 antibodies. You would be able to profile both genotype and phenotype information from the same workflow. So those are the two main assays, and the sample prep associated to both the workflows are slightly different. So I will go over those two first. So for the DNA-only workflow, in terms of sample preparation, there aren't a lot of steps involved. Users can start with either cryopreserved cells or fresh cells, and then depending on how you cryopreserve, usually we recommend at least having anywhere between 50 to 90% FBS with media and 10% DMSO, so that they're very viable when you thaw these cells. For thawing, stick to standard thawing protocols where you could thaw your cells at 37 degrees, followed by adding thawing media, etc. Whereas if you're starting with fresh cells, you could go ahead and start centrifuging your cells, and we require you to wash away all the remaining media and FBS and other materials. And after a couple of centrifugation steps, once you have these cells that are in PBS, we would like you to proceed to count your cells. Once you're counting the cells, there are a few tips that I will provide in the next couple of slides. How do you assess the quality of the cells? How viable the cells are? Are these cells, very well suspended? If they're aggregated, what do you do, etc. So if you're starting with, viable cells, intact cells, the sample prep process is as simple as just a couple of wash steps. However, if you're starting with a frozen tissue section, we have a nuclei isolation protocol, which I will discuss towards the end of this talk, where we have a protocol that has been optimized to isolate nuclei efficiently from tissue sections. And then the goal is that either if you're starting with intact cells or nuclei, you can get up to 100,000 cells or nuclei, which is required as input to start your Tapestri workflow. Tapestri workflow, basically just as a quick summary or a brief summary, we capture cells in oil droplets and we amplify the content using targeted panels. So the first part is to encapsulate these cells. And in order to encapsulate these cells or nuclei isolation, we need a clean nuclei suspension or cell suspension, about 100,000 in number. So comparatively for DNA plus protein workflow, one of the biggest differences is that once you have your cells thawed and ready, we have a staining step during which we stain the cells with our antibody mix or antibody panel mix. And in order to do that, you have a requirement to start with about 2 million or more cells, so that after your washing and thawing, you have about 1 million cells, which we stain with our antibody tagged oligos. And then after that staining step is done, there are an additional washing steps to remove all the untagged antibodies, etc. And then once you finish that processes, again, it's very similar in terms of starting amount. You require, again, about 100,000 cells that will go into the Tapestri workflow for the encapsulation step. So sample types that are compatible with the platform. For the DNA workflow, you could start with cell lines, both suspension or adherent cell lines, cryopreserved PBMCs, bone marrow cells, And all these type of cells are compatible, or sample types are compatible with both the DNA workflow as well as protein workflow. However, when we're working with solid tissues, we have validated the workflow mainly for DNA only. The protein aspect of things, where it's very important that you're starting with viable material, and so that you have intact membranes where the staining of the antibody oligoconjugates happens, and so that is critical. So we haven't validated tissue sections, isolations, etc. for the protein workflow yet. For DNA only, you can proceed with OCT embedded tissue sections or frozen tissue biopsies, or if you have other nuclei isolation protocol, in addition to the one we will discuss today at the end of the talk, feel free to use additional nuclei isolation enrichment protocols if you can get clean nuclei and you're ready to proceed to the Tapestri workflow. What we don't support, whole blood cells, where you have a lot of red blood cells in your samples. We recommend you to deplete the red blood cells or do additional, phycol extraction or etc., where you can get your white blood cells. FFPE is not currently supported in the platform. And we have shown some early proof of concept data from using just non-viably frozen just cell pellets seems to have worked with the platform, and a light fixative like PFA 1% or 2% seems to be also working well with the platform. So now addressing cell viability, one of the key important thing to notice here is that for the DNA only workflow, if you're visualizing your cell sample, here I'm showing you an automated cell countess, you want a very good dispersion where you don't have much aggregation of cells. And here we're showing you a simple staining for cell membrane integrity using trypan blue. You can see the viability is about 69%. However, for a DNA only workflow, this sample is good enough to proceed. For a protein workflow, this is a little bit below the recommendation of viability. You need more than 80% viable cells to conduct the DNA plus protein workflow, because we need to stain these cells with our antibody conjugate pool. So additional dyes that you can use to stain your cells, other than just the cell membrane integrity dyes as trypan blue dye DAPI, you could use enzyme activity based dyes such as calcine based or acridine orange, etc. So the cell size is also another important question that comes up. Tapestri platform has validated cells that are ranging from 2 microns, primary cells, all the way through large cell lines up to 30 microns, or some macrophages that are up to 30 microns. That's like our upper limit. We haven't validated cell sizes or fibroblasts or something that are larger than 35 microns. So 2 to 30 micron is the ideal range for cell size. And now coming back to just the DNA only workflow, continuing from what we discussed on the last slide, the question of viability. If you have, cells that have intact nuclei, even if your viability is a little bit poor after you thaw your, frozen samples, for the DNA only workflow, it doesn't impact the performance of the, the workflow. Here we are showing you an example where on the top left corner, these are several different samples of thawed PBMCs. And after thawing, we're showing you a percentage viability. Viability has been as low as less than 10%. Sometimes it's as high as 90%. But regardless, after the workflow is conducted, all our quality metrics that we are showing on the right-hand side, we don't see a drastic difference if the cells are a little bit lower in viability, as long as you're only doing the DNA only workflow. So again, as long as you have intact nuclei, the DNA only workflow seems to be, very forgiving. Now, additionally, there are extra steps to get a much cleaner sample material. You can do a FACS sorting or a MAX-based approach where you can clean your material before you start the workflow. Here I'm showing you some approaches where you could remove debris, or if you think there are dead cells or doublets, you could use a FACS approach, and you could clean up your samples before you start your workflow. If there are any questions right now, I could answer them so far. Um, uh, Kelly, are you online as well?
Yes, hi. I'm fielding the Q&A, and I think we'll wait till the end of your seminar in order to field all the questions.
Absolutely. All right, so we'll continue for now, and we will have a 10-minute Q&A towards the end where we can answer all the questions. So now, just as a summary of what we have talked so far, the code says going garbage in, garbage out. So we have to make sure we're starting with high quality sample input in order to get really good data output. So these are some of the things to pay attention to, that the sample at the end of the sample prep, you have a sample that are free of dead cells or other debris, etc. And always, if you have cryopreserved samples with high viability, that will be the first recommendation. If not, if you are doing the nuclei isolation protocol, we will discuss that towards the end. And here are some additional tips where if you wanted to disaggregate your cells, you could add BSA or EDTA, etc., to your cell buffers. And just in summary, just to compare the DNA only workflow and the DNA and protein workflow, the chart here shows your starting number of cells required. For the DNA only requires about 500,000 cells, whereas for DNA plus protein, you need upwards of 2 million cells to start the workflow. That is because you have a staining step where you need about 1 million cells to stain with your antibody oligo conjugate cocktail pool. And then once you're starting the Tapestri workflow, where you input your cell materials into our microfluidic cartridges, the amount is 100,000 cells. That does not change. All right. So the last part of the talk, I want to focus on the nuclei extraction workflow. So this is a user guide released by Mission Bio. You can download that from our support website. So I'm going to go into the details about how the workflow, or what are some of the tricks and tips where you can isolate nuclei successfully from your tissue sections. So we're on the right-hand side, we're showing you an example image of an OCT slide where, let's say you have your tissue. We recommend that if you have a thickness of about more than 20 micron, 20 to 50 micron slides will be great. And the example here is that based on the thickness of your tissue type, you can use a reference slide just to estimate what could be the length and the width of your section. And let's say you have a 20 to 50 micron thick tissue, we can calculate the tissue volume just by multiplying length times width times thickness. And we have learned that for a decently nucleated section, about an 8 millimeter cube is sufficient enough to produce upwards of 500,000 nuclei. However, that varies between sample type. Sometimes you have a very less nucleated section, so you may need to have a little bit more larger tissue section, or you could use a couple slides instead of one slide. And then the goal here is that once you go through the nuclei isolation protocol, at the end, you have clean isolated nuclei, and a sufficient number is about more than 500,000 if you can, or as low as 100,000. And so that is the goal here. So we will go through the steps involved, and I will highlight some of the important steps of this protocol. So on the left-hand side, the process starts once you have your OCD embedded or tissues or snap frozen fragments. You take these sections and you treat them with our tissue lysis solution. Our tissue lysis solution is a cocktail mix that contains trypsin, collagenase, dispases, and some buffers. And after you treat the tissue sections with our lysis solution, as a reference, on the right-hand side, we're showing you a picture of an example tissue section and how we're going to do the workflow. We take a Petri dish, put it on top of dry ice. After we take one of these sessions, we put them in the Petri dish, add the tissue lysis solution on top of that, and once we wait for a couple more minutes, and once the tissue lysis solution has been incubated, we slice them up into a few pieces, and then we will do a quick centrifugation. And before we do that, we inactivate the trypsin using a stop solution, and we use a strainer to make sure before we spin the nuclei down, we're removing all the the unwanted materials, and only the clean nuclei are collected. And once the nuclei are collected, it will look like the picture onto the right. For an example here, we're showing you a melanoma section. So melanoma cell or tissues are enriched with melanin, and that is the brown color that's shown in the representative image. And your nuclei could look a little bit brownish. But the workflow works well even with melanoma tissues. The melanin doesn't interfere with our downstream workflow. So once you have your nuclei, one additional step that we have incorporated into this workflow is that we have a fixation step. To this nuclei, we add a methanol acetic acid solution. The reason why we do that is that it seems to improve the structural integrity of the nuclei, and it's minimizing clumping. And so once we do that extra fixing step, we will do one more centrifugation, and then followed by an additional strainer filtering step. And once that is completed, you visualize your nuclei under a microscope and using a hemacytometer. And so you stain your nuclei, we recommend with the DAPI dye. And then I'm going to show you some tips and tricks how a good isolated nuclei at the end of the sample prep should look like. But before that, these are the validated tissues that we have done with our workflow so far. It ranges from a lot of tissue types, colorectal, pancreatic, a lot of melanoma samples as well. And we have shown successfully the protocol work with different tissue types. So coming back to the point that I was making, on the bottom left, an optimal sample prep output will look like this, where you have nuclei very well separated, not a lot of aggregation, and the DAPI shows clear staining for your nuclei. Sometimes the tissue lysis step, where we digest away your tissue fragments, it requires a bit more optimization depending on your sample type. Sometimes you may over digest your prep, and if a situation like that happens, what we're pointing here all the way under the over digested picture, you could see these strands of nuclei, and that is not really good for the platform. You want the nuclei membrane to be intact. So you have to watch out for that. And then you may want to reduce the time of digestion. Sometimes you get under digested products as well. So here we're showing you an example. You could stain your prep with a cytoplasm staining dye, as well as a membrane staining dye, where you could see that how your nuclei prep has been digested. So you definitely wanted to optimize the workflow and then adjust your digestion times and cleanup times accordingly. The user guide goes into a much more detail for these tricks and tips, but this is basically how the workflow is constructed. Lastly, we have our Mission Bio support website. Please visit the missionbio.help, where we have sessions for sample prep all the way through analysis. So there's a lot of useful documentations on the website as well. And lastly, again, just as a reminder, next Tuesday, we have Dr. Ross Levine giving a talk and use how he has been using the Tapestri platform, addressing questions, etc. Please, follow the link and definitely sign up for that webinar. And then for the next Thursday support office hours, my colleague will be continuing this discussion, and we'll discuss what we do on the day one for the Tapestri workflow. So now I guess I can take questions that have come up, and then so Kelly, should I just read the questions and then answer them, or would you like to ask the questions?
I'll ask the questions. So,
Okay.
Jose, thank you so much for a really nice seminar on how to do sample prep and nuclei extraction. I think it's going to be really helpful for our customers. So we got a lot of good questions in the chat box. And so for our first one, for cryopreservation, have you found slow freezing to be necessary? They found it to be necessary for single cell RNA-seq.
Yeah, I mean, great question. So right now, we do recommend slow freezing, and you can get these boxes that are made for it in terms of reducing the temperature at 1 degree Celsius per second. That's what our recommendation has been. We haven't tested extensively what happens with or without, but we strongly recommend slow freezing for all the workflow. For generating, because once you thaw your cells, we definitely want highly viable cells. So that's definitely a recommendation.
Great, thank you. You mentioned that whole blood wasn't compatible. What method would you recommend for red cell lysis to deplete red cells?
So there are kits and just a cation treatment or just a buffer treatments where you can pop the red blood cells. So any standard protocols just to do a quick popping of the red blood cells and cleaning it up, or you can just do a FICO based approach where you can treat your cells with FICO extraction and get the PBMCs or other whole white blood cells out of it. So the red blood cells itself could potentially interfere with the chemistry. However, again, this is just a recommendation. We had customers mainly working with FICO extracted PBMCs or BMCs instead of working with whole blood cells. But yes, please entertain the options of depleting RBCs by popping them with a salt solution, ammonia-based, or I can't remember. But yeah, there are some couple of protocols that we can share with you. Hello?
Thanks, Jose. We've got probably a couple more questions, if you don't mind.
Sure.
Okay, let me pull those up.
Hi, I can ask the next one. So you mentioned that viability has to be greater than 80% for protein, but not for DNA. Can you explain why that is?
Absolutely, good question. So for the DNA workflow only, we're just amplifying content and the material for the content comes from the nucleus. That is our template. For the protein workflow, the cell membrane is very required because these protein or AOCs that we call are the antibody oligoconjugates are tagging surface markers. So if when I mean viability, we mean cells that have intact cell membrane, and that is critical. Otherwise, if you're staining with our AOCs, there could be a lot of background staining of these, potentially compromised nuclei membrane, cell membrane cells or dead cells. So in order to avoid that, we need high viability for the DNA plus protein workflow.
That makes a lot of sense. Thanks for the clarification.
Absolutely.
All right, another question. Can you use the same cell surface markers for FACS enrichment and use it for the DNA plus protein Tapestri workflow?
That's a great question, right? So here's the approach that we usually take. For your for your Tapestri AOC pool, we do let you know what the clones of the antibodies that we are using. And when you're doing FACS enrichment, we generally, strongly recommend you do not use the same clone. And in an ideal case, it will be better if you're FACS sorting based on a different marker that is not part of your AOC pool, just to avoid any steric hindrance or etc. Another approach that people have been using is just doing a forward scatter or just using a regular FACS not to just enrich for any markers itself, but just to get a population of your interest. But if you have to use a marker, just in the best case scenario, don't use a marker that is part of your AOC pool that we provide. If you do, we haven't validated, definitely do not use the same clone. Use a different clone. That may be the best case, but again, we haven't validated that approach. So that's the consideration, is that there might be some steric hindrances if you use the same marker for FACS as well as in your antibody oligo pool to conduct the workflow.
Thank you. Does Mission Bio have a validated protocol for mitochondrial DNA isolation?
Again, a great question. So we do not have a mitochondrial isolation protocol right now. It's, in early R&D, we have entertained, mitochondrial isolation, but it's not something we support at the moment.
All right, just a few more questions here. Can I use Streck cell preservative of my samples prior to use on the Tapestri?
Yes, Streck tubes has been something we have been testing as proof of concept. So we have seen some early data where the performance seems to be working. So Streck tubes being a mild preservative in the Tapestri workflow itself, how we lyse our cells after they are encapsulated, it's a pretty long 1 hour step. So these mild preservatives usually gets cleaned up during that, and you get access to your DNA. So PFAs, 1%, 2%, Streck tubes, early data has shown it seems to be working, but we haven't shared any validated data set, but it's an ongoing work currently.
All right, and then a final question. Have you validated nuclei extraction from heme samples or cell lines? If so, have you compared data from whole cells versus isolated nuclei from the same sample source?
Good question. So I will have to check back into our support materials in terms of we have done a direct comparisons. We have done independent experiments of both those categories, but I am not sure if we have a direct comparison, but I'll definitely go back into our support website and let's see, I'll check that if we have that direct comparison. Not that I know of right now, but we have done it independently.
Great. Well, Jose, I'd like to thank you on behalf of all of our attendees and Mission Bio community. We really enjoyed your talk on sample prep, and then we're very much looking forward to next week's series, which again, we have Dr. Ross Levine from Memorial Sloan Kettering talking next Tuesday, followed by another installment of Office Hours on Thursday. Thanks again for joining us, and we'll see you next week.
Alrighty, bye-bye, guys.